Welcome to my lesson on transpiration in plants.
We're going to look at the process of transpiration and how environmental factors affect it, and practical investigations using pitometers. Transpiration is the loss of water vapour from the upper parts of the plant, mainly through the leaves through the stomata. The transpiration stream is the flow of water through the plant from the roots to the leaves. The water moves through the xylem primarily as a result of evaporation from the leaves. The cohesion between water molecules and the adhesion of water molecules to the walls of the xylem create an unbroken column of water. There is also root pressure. When water enters the root from the soil, it creates a weak pushing-like force, which helps the water move upwards through the plant. In some small plants, root pressure can actually push some water droplets out of the plant, and this is known as guttation. Root pressure on the whole only has a small effect on the transpiration stream. Approximately 99% of the water absorbed from the soil is lost by evaporation from the leaves and stem. The water moves passively down the water potential from the roots to the air surrounding the leaves. The gradient of the water potential is the driving force behind the movement of the water. The benefits of the transpiration stream is that as the water evaporates from the leaves, it cools the plant. The water transports minerals up the plant from the roots. The water makes sure the cells remain turgid, and it also supplies water for cell elongation, e.g. phototropism, for growth and for photosynthesis. Potometers are used to investigate the rate of water uptake by a plant. There are lots of varieties of them around. This is one that SAPS recommends, and they have a good video explaining how to make one and set it up on YouTube. We can then use this to investigate the influence of environmental conditions on plant transpiration rate.
You can use pitometers to look at how different environmental factors affect the rate of transpiration. First of all, you can set it up at room temperature. Then you can use wind, so you could use a hairdryer or a fan, and then high humidity by putting a plastic bag over the leaves.
The apparatus is set up underwater in a bowl. The chute is cut underwater and inserted into the apparatus whilst underwater, and an airtight seal has to be established in the apparatus. The leaves then need to be dried, otherwise the first lot of water to evaporate will be the water from the bowl, not the water evaporating out of the stomata. This is a classic pitometer found in schools. The cut chute is inserted into a tube or cork at one end. An air bubble is introduced into the end of the capillary tube, usually by dabbing it with some tissue paper. Then you can either measure the distance travelled in a set time or the time taken to travel a set distance. Repeats need to be carried out to identify anomalous results and to make results more reliable or repeatable. It is best to do repeats by keeping the apparatus set up the same each time. The easiest way to do this is to open the water reservoir between trials to reset the bubble to its original position. The volume of water taken up can be calculated if the diameter of the capillary tube is known, by using the equation for volume of a cylinder, pi r squared h, where h is the distance travelled. Then rate of transpiration can be calculated by dividing the volume by time. Remember, this is only an indication of rate of transpiration, as some of the water is used by the plant cells for other things, such as photosynthesis. So just to go through the main points you need to remember when setting up the pitometer. Make sure you cut the chute underwater. Insert it into the end of the pitometer underwater, making sure you have an airtight seal. You can then take the pitometer out of the sink or bowl and dry the leaves to make sure that any evaporation of water is out of the stomata, not off the surface of the leaves. Introduce an air bubble into the end of the capillary tube as you use this to track the movement of water. Record the distance travelled in a set time. You can then use this to work out the volume of water lost by transpiration in a set time, and hence the rate of transpiration. Remember repeats, at least 3 results for each new environmental condition.